Welcome students to Genetics Summer 2018. I shall be your instructor. My job is to make sure that each lecture you folks have the necessary resources to do well on the topics discussed. So without wasting too much time, let's get started with Chapter 1. Chapter 1 is a basic introduction to genetics. Many of the topics covered in Chapter 1 will be expanded upon in future chapters in far more detail. So Chapter 1 can be considered a two-part chapter, where the first part is a history of genetics, and the second part is a review of information to come in future chapters. What we like you to take away and bring to the first lecture is all the information that's highlighted in various colors on these PowerPoint slides. So this is slide number 1. And it talks about the position of genetics currently. And you can see here, students need to remember that in the past 10,000 years, humans have been performing knowingly or unknowingly genetic experiments in terms of selective breeding of some familiar and vitally important food substances such as the cereals mentioned here. However, these previous manipulations were rather rudimentary and required very little understanding of genetics. Today, technology has progressed to the point where we can now manipulate any component of the genetic environment that we wish within the technology framework that we have. To support the statements on the previous slide, we need evidence, and the evidence is presented here in panel 1.1, part A, by showing you that we have reliefs, etchings in stone, showing what appears to be humans manipulating plants. And in panel B, we can see examples of the progress that humans have made, here with an example of the modernization of corn. The next couple of slides give us a historical perspective on how genetics, modern genetics, came into fruition. The authors begin with a story starting in the 1590s with the development of microscopes, very early magnifying glasses, which eventually led to refinements leading to the discovery and reporting of the nucleus inside cells in 1831. Just a few decades later, an amateur botanist who happened to be a monk named Gregor Mendel published a scientific paper where he tried to explain how pea plants inherited certain characteristics. And we're going to look at those in a future chapter. And this took place in 1866. Unfortunately, Mendel's work was mostly ignored until it was rediscovered independently by 3 scientists whose names are not important, mainly because I haven't coded them up here on this slide, nor will you be tested on those names. In the very early years of the 20th century, in 1901, a paper published by Giraud, a physician in England, described a genetic disorder where the urine of the patient was discolored, basically black, a medical condition known as alkaptonuria. Indeed, other scientists studying the same condition later, such as Bateson, recognized that it was a rare condition and that it repenetrated families in a recessive manner. Next, the textbook tries to define some of the key terminology important to all genetic students. So one of the things that you must remember is the definition of what a gene is. For now, the definition of a gene as the physical unit of heredity is good enough, but later on we'll define that in the context of more recent findings. What we require from you as the student is to have a solid definition of what a gene is by the end of the course. Likewise, the definition of chromosomes, according to Chapter 1, is that they are long molecules of double-stranded DNA associated with proteins, and they contain genes at various positions along their lengths. Most sexually reproducing organisms, plants, animals, and fungi, usually have 2 pairs of chromosomes of the same exact type known as homologous pairs, or homologs as short. And because they are almost identical, they carry the same genes at the same positions, therefore specifying the same traits. So the word trait can be substituted for the word characteristic. So you need to become familiar with this terminology. What we suggest you do is start a glossary of your own where you add from your definitions throughout this course. We shall be devoting an entire chapter to understanding the genetics of mitochondria and chloroplasts later on in the course. But for now, what you need to know is a recall of information that you have learnt in the past, that both animals and plant cells, they contain organelles called mitochondria, but only plant cells contain chloroplasts. The importance of this information is relayed by the last statement here, that each of these organelles, the chloroplast and the mitochondria, they do contain one or more copies of their own DNA. So they do contain their own circular chromosomes, and those chromosomes have on them a certain number of genes important in manufacturing material for the cell. And the way that those DNA molecules within chloroplasts and mitochondria are then separated and replicated into future generations is something that you have to learn. Contrasting the situation between different forms of living cells is one of the requirements of this course. This slide talks about chromosome replication, which we'll talk about more in depth for the next lecture, chapter 7. But for now, it's important for you to know the differences between different branches of the tree of life. So bacteria and archaea, they have normally a single chromosome, which is circular, double-stranded DNA, and it's found outside of any organelles. So it's called a nucleoid, and that replicates through binary fission in the cell, and we'll talk about that in the future lecture. But in eukaryotes such as us, we have multiple linear chromosomes that are present inside a nucleus. So the geography is different. Therefore, the question is, are the mechanisms of chromosome replication different? So you need to know the answer to that. Sexually reproducing organisms produce single cells through meiosis called gametes. And the gametes have half the amount of DNA as the cells from which they arose. The names of the gametes are sperm and eggs in animals, and pollen and eggs in plants. So the word pollen is used for plant male gametes, whereas sperm is for animal male gametes. With respect to genetics, the most important thing is that the passage of genes between parents and offspring is transmitted through predictable patterns that we either fully understand or are trying to understand. Here are 3 further definitions for you to learn on a permanent basis. You should be able to tell the difference between a phenotype, a genotype, and the application of the word alleles. So a phenotype is basically the observable outcome of genetic mating of an organism. So somebody with brown hair. The genotype is the genetic makeup of that organism. Do they have 2 genes for brown hair or just 1 gene for brown hair? Alleles are straightforward. These are alternative forms of the same gene. So brown hair versus black hair versus blonde hair versus red hair would be nice examples. So the passage of genes from parents to offspring is called transmission genetics, and is one of the 3 branches of genetics that we're going to have to learn this semester. So here comes the history lesson, the main learning objective of chapter 1. Any names that you see highlighted in different colors other than black, you have to then learn. So the experiments performed by Avery, MacLeod, and McCarty have always been cited as being very important in identifying DNA as the substance through which material is inherited by offspring. Prior to this, people used to think that protein was the main molecule that was passed from parents to offspring. But their work pointed the finger at DNA. These experiments were the beginning of the molecular analysis of genetics. So we call that the other branch, the second branch of the three that we have to learn this particular semester, called molecular genetics. These experiments subsequently led to the greater understanding of how the structure of DNA imparts the information content, and then how DNA itself is replicated so that each of the two daughter cells, in the case of mitosis, and the 4 daughter cells in the case of meiosis, and the 2 cells in the case of binary fission, each receives the appropriate copy of the DNA molecule. Other than DNA replication and its structure, students need to understand the mechanisms underlying transcription, the copying of DNA into RNA, and translation, the transfer of information from RNA into proteins or polypeptides. At the same time, in translation, the genetic code needs to be learnt by us, and the history behind how it was deciphered using the technology at the time. And subsequently in the 1970s, genetic engineering came to the forefront with our first attempts at cloning genes from cows, bovine growth hormone, and then the subsequent development of the technologies around that has implications for us today. So if you listen carefully so far to this video, you will see that I am trying to tell you guys a story, and learning through storytelling is one of the most effective ways in which somebody can retain knowledge long term. One method to keep these stories and memories alive is to become the teacher to another student. So teach somebody else what you think you know about a particular subject or topic, and you'll have a chance to rekindle your stories and to double check any areas that you may have misperceived or forgotten. The next set of slides that we're going to cover talk about individual regions of understanding that you should be familiar with that may need some reinforcement, but it's your job to understand exactly what the definition of each component is. So if somebody asks you what is a genome, could you describe that? Well, here's a definition from our textbook. A genome of an organism is the complete set of genetic information carried by that particular species. Genome analysis is a pretty recent development in the field of genetics. And in the 1980s, the tools were developed to enable scientists to study and then compare using sophisticated computer programs the genomes of several organisms. And since then, this has flourished into an easy to generate set of data. So now we can generate data from lots of organisms quite rapidly. 2001 stands out as a landmark year because in 2001, the first draft of the human genome was published by collaboration amongst many laboratories around the world. So that was a massive project costing $3 billion and lasting 10 years. Understanding the past is just as important as understanding the present and predicting things into the future. So one of the things that we want to know is, where did present day life come from? Where did we come from? And studying certain features of our genes and our chromosomes and our biology has allowed us to have an insight into common ancestors, the ancestors of present day creatures. And the most important ancestor that we've been seeking was the last universal common ancestor, Luca, of all life forms. This, we believe, were the first cells or the first species to have colonized planet Earth, either from space or by evolution in the oceans on the planet itself. The 3-domain model of life is credited to Woozy and colleagues who used ribosomal sequences, ribosomal RNA sequences, to establish the phylogenetic relationships amongst living creatures alive today. So phylogenetics is a way of looking at the phylogeny, the actual relationship between the branches of the tree of life involving species and their genetics. Because RNA is a molecule of inheritance in some creatures and an important molecule of information in all other creatures, comparing those sequences gives the computer enough information to rearrange the species in order of similarity. So closely related species have more similarity in their ribosomal RNA genes than do more distantly related species. By feeding this information into computers, we can then generate trees like this one, which suggest to us the relationship between different branches of the tree of life. A computer doesn't know any bias, so by feeding it information and asking it to rearrange things mathematically based on A's, C's, U's, and G's, it can then generate these trees without any bias. So this is where the information for the three branches of the three domains of life has originated. Incidentally, the picture has been complicated, blurred, as you may say, by the migration of information between branches that has taken place in the past and may be taking place currently. So you can see chloroplasts, we believe, have an origin in the bacterial domain, and they then make their ways as organelles that impregnate eukaryotic cells. And the same thing with mitochondria. But we'll talk about that with the theory of endosymbiosis a little later on. As you should have learned in the past, all living things are made of cells, so the cell theory of life. And within that theory, the definition of life itself tells us that all living cells without exception, do contain DNA as their molecule of inheritance. Learning the structure of DNA is important for students. So every time you're asked to draw DNA under normal circumstances, you have to remember that it's a double-stranded molecule consisting of two separate strands wrapped around each other in a helical fashion. Incidentally, just for the sake of completing the story, viruses are not considered living because they're not made of cells, but they have evolved to carry RNA as their molecule of inheritance rather than DNA. Because life started a long time ago and then branched into the three branches, the three major branches, the domains that we spoke about a few seconds ago in this particular slide, what did living things look like at the beginning? How did they, when they weren't divided into three domains, but were a single domain, what did they share in common? Well, our research suggests that DNA replication is a process that was invented very early in the evolution of life, probably around here on this black line somewhere. And then that process was passed more or less unchanged to the three domains of life. Not just DNA replication, but also the way that information from DNA is extracted and placed into RNA, and also how the information in RNA is then fanned out to the ribosomes to be converted into proteins and polypeptides. Thus, one would expect very few changes in these biochemical processes to have evolved since those early days. And that's true. So if you learn DNA replication in bacteria, with very few modifications, you can apply that to the other two domains of life. In conclusion, we can say that modern genetics has three branches itself, just like the three domains of life. So that's an easy relationship to remember. And the question then becomes, what are those three branches of modern genetics? And we mentioned two of them earlier. The first one was transmission, inheritance of genes from parents to offspring. So that's called transmission genetics. Some books call that Mendelian genetics in honor of Mendel. The second mechanism we spoke about earlier too is molecular genetics. And the new one that we haven't spoken much about, but we did mention very briefly when looking at the domains of life, is how one species is related to others through evolution. and that's called evolutionary genetics. And that deals with relationships between organisms, populations, and species. And we'll look at that later in the course. For the next 30 slides, 30, I'm going to go through rapidly, explaining my interpretation of the slide, but most of you should be familiar with the contents from previous knowledge. So rapidly then, knowing the structure of DNA elicits a way that DNA can be replicated. So DNA contains an inherent feature to copy itself. So the structure of DNA is very important, and you'll be asked to learn that in detail so you can redraw it and replicate it multiple times. And from that, we can then extract and extrude information other than just carrying information, also a mechanism for replicating and passing on identical copies of that information to future cells. So where do we start? Well, we start back in 1953 when Watson and Crick published a paper revealing the structure of DNA. That work was not their own. All they did was put the pieces together while sitting around in pubs. But still, regardless, they do get credit. The bulk of the work was done by other scientists who published their work at the time. So one of the most famous was a young American lady working at King's College London, and her name is Rosalind Franklin, and she unfortunately died before the Nobel Prize could be awarded for eliciting the structure of DNA. So she doesn't get that, unfortunately. But her work was very, very important. So in recognition of that, here's a picture of Rosalind Franklin taken when she was doing research at King's College. This represents one of the most important pictures that was used by Watson and Crick in determining that DNA was a helical structure and that the two strands of the DNA were running in opposite directions. Now, don't ask me how this works. It's a very sophisticated science. I could never tell you. But this is what they elicited from that picture. And at the same time, another scientist, a French scientist, Erwin Chargaff, he was doing biochemical analysis of DNA from different species taken from zoo animals. And he came up with this formula here, that the number of bases along a particular double-stranded helix is always equal to the number of thymines, always. And at the same time, the number of cytosines is always equal to the number of guanines, always. So this is Chargaff's rule. This rule has never been violated at all in any species that uses DNA as its molecule of inheritance. So from this, Watson and Crick were able to conclude that base pairs may exist in a complementary fashion, where if there's an A on one side of the double-stranded helix, the other side must contain a T, and likewise with the G and C. We now know that if you extend the analysis beyond zoo animals into any other living creature, the same rule applies across the board. So Chargaff was instrumental in elucidating the complementary base pairing of DNA. This figure here shows the rudimentary structure that Watson and Crick devised from the analysis of other people's work. So this was the very first DNA double-stranded molecule to be put together using cheap implements available to them at the time. I did name the 4 nucleotides. That's important because there's something called a nucleoside, which we'll talk about later. But the DNA molecule is made of building blocks known as nucleotides. There are 4 nucleotides across the board. They are adenine, guanine, thymine, and cytosine. And each nucleotide is linked to its neighbors through something called a phosphodiester bond. A phosphodiester bond. Now, if you go to this figure here, you'll see that phosphodiester bonds are present right there in this yellow region of the strand of DNA. And there'll be another one down here and another one up here, right? So understanding that in the future is going to be paramount in our understanding of DNA structure. But for now, just remember what a phosphodiester bond is and how to spell this. Subsequently, the next thing that we need to know is that hydrogen bonds hold the two strands of DNA together. Hydrogen bonds hold the two strands of DNA together. So again, referring to a figure, and figures are very important, we can see here that if this is one strand of DNA running up and down, and this is one nucleotide, and this is its complementary nucleotide on the other side, that will be part of another strand that runs up and down here. The two halves are held together by these dotted blue lines. And these dotted blue lines are hydrogen bonds. We'll talk more about those in the future too. Now, the only way that you can get the hydrogen bonds to form across that gap between the two strands is if one strand runs one way and the other strand runs the other way. And that is what we call antiparallel. So this strand here in blue, it goes from the top, which is the 3' end, to the bottom, which is the 5' end. The red strand starts at the 5' end and then migrates down to the 3' end. So the two strands, when you compare either end, are not identical. They're running in opposite directions. So we have to learn that in the future. The next thing is that we need to understand how DNA replication takes place. So here we have the history of DNA replication, and in Chapter 7 we'll go into more details about the biochemical mechanisms by which DNA is replicated. So the important thing to understand is that each strand of DNA contains information that will allow you to predict the opposite strand simply by looking at the information in the first strand. And the scientists that were responsible for elucidating this information were Mendelssohn and Stahl. And they demonstrated that DNA was replicated in a semiconservative replication. That means the two strands of DNA separate and new strands are built on that particular backbone. So one of the things we'll get you to do is to understand the three contenders for DNA replication. One was the conservative method, the other one was the disruptive method, and the third one is the semi-conservative method. In all natural systems, DNA replication has to begin at some place, and that place is not random. It's always defined by evolution, and it's called the origin of replication. It's the place on the DNA molecule where replication begins using enzymes. And as we said earlier, DNA replication requires help, and the help is provided by these proteins, and the name of these proteins happens to be DNA polymerases. Why? Because what they're making is DNA, and how do they do it? By polymerizing the nucleotides into DNA strands. Unfortunately, these enzymes have evolved to only work in one direction, which is 5-3 direction. So DNA is always replicated backwards. It's read forwards, but replicated backwards. And this is something I'll keep telling you until it fixes in your head through trial and error. So here's a figure showing one replication fork, and we'll talk about replication forks in the future. At this replication fork, we have the original strand, which is separating here in red, from the other strand, which is in blue. And each strand is then used as a template to make a fresh copy of DNA using nucleotides. So this is one of the important goals that we have to learn, and there's a lot of questions on this on midterms and the final exam. This then raises other biochemical questions, such as can information in DNA skip the RNA step and end up going straight to protein? And the answer is no. There's no known mechanism where DNA, under natural conditions, can be directly converted into protein. So if it can't be converted directly into protein, what does it have to go through? Well, the answer is it has to go through RNA. So RNA is a very, very important intermediary between DNA and protein. And what you'll learn in the next few weeks is that not all information has to go all the way across to protein. Some information can stay within DNA, other information is converted to RNA, and then some information gets converted to protein. These arrows are one way, and they then tell you how information flows in biological systems. And this is called the central dogma of biology. A dogma is an unbreakable rule that we have established. This slide reinforces the fact that RNA molecules can fall into different classes or types. The most important class that you know about is messenger RNA, and messenger RNA is used to convey information to make protein. But in addition to that, we have RNA that forms a structural function. So this is information, and this is structure. So the ribosomal RNA physically forms part of the framework of ribosomes. And at the same time, other classes of RNA, other types of RNA, do other jobs inside cells. And in this case, transfer RNA is used as a transport vehicle for bringing in the amino acids to the ribosomes. Again, we're going to learn all this in very, very fine detail in subsequent chapters. But for now, we also need to know that scientists have discovered other types of RNA that are important in controlling the behavior of cells. So microRNAs, these are very small RNAs, 20, 30 nucleotides in length. They are very important in controlling the behavior of the central dogma, how information is passed from DNA through to other molecules. And we're going to have to learn that in a lot of detail in the future. What you see here is that since then, many other types of RNA have been discovered. In fact, if you look on Wikipedia, you have more than 40 different types, 40 types of RNA, and we're going to learn most of those by the conclusion of this course. And then finally, viruses upset the horse and the cart model because they can do strange things in that they can convert RNA back to DNA before it travels down the central dogma. But that's an exception. Here we have two more terms for you to learn. When DNA is separated into two strands, each strand can be identified using a label. So the strand that's used for making RNA from DNA is called the template strand. The other strand, the one that's not used by the proteins for making RNA, which just hangs around in the vicinity, is called the coding strand. Strange but true. And also you can also have other labels. So if this is the template strand, the other strand can be called the non-template strand. And if this is the coding strand, the previous strand can be called the non-coding strand. And I'll explain all that with a diagram in class. So here we have a summary of transcription taking place. This brown object is an enzyme, and this time it's called RNA polymerase, not DNA polymerase, because the question is, what is it making? It's making this green strand of nucleotides, and that's RNA. So this is RNA polymerase. You can see the two strands of DNA being separated temporarily, and this enzyme will move in this direction because it can't move in any other direction. And as it moves in that direction, the DNA on the bottom strand, this will be the template strand, is then not copied directly but interrogated to discover what that base is. So if that base is, for instance, a C, then opposite that you will get a G coming in next, as will you in the next position. So this system is supposed to be fail-safe and very reliable. One of our favorite questions in genetics and cell biology is for students to be able to compare and contrast the structure of RNA and DNA. And as you may have heard me, I was very careful in using uracil when I was talking about the structure of RNA and not mentioning thymine, because thymine is not found in RNA, it's only found in DNA. But RNA consists of a different sugar too, not just one of the bases being different, but it has a different type of sugar that it uses to form its strands. And we'll learn that in the future. And as we just mentioned, RNA polymerase is the enzyme that copies DNA of all living things into a single strand RNA molecule. So the word regulation can be translated to mean control. So whenever you see regulation, substitute that with the word control. So control of transcription. How do you control transcription? Well, transcription is a process that has to be carefully controlled. You only need to turn on genes when you need them and turn them off and keep them off when you don't need them. So we have switches called promoters that are present normally at the beginning of a gene, and when the promoter is available, it will then bind the machinery, the RNA polymerase, to perform transcription, but not always at the same speed, at the same rate. Sometimes the promoter speeds up, other times the promoter becomes more silent. And that depends on the conditions in which the cell finds itself. And then when the transcription machine gets to the end of that gene, it needs to be able to identify when not to go any further, and that's called the termination region. And the termination region, again, is a sequence on the DNA that tells that machine, stop here, don't go any further. Lastly, we have some very peculiar arrangements of genes that eukaryotes have taken to the next level. And so our genes, most of them contain regions called exons and other regions called introns, which have no known function, and they are actually removed before mature messenger RNA is produced. Combining all the knowledge from the previous few slides together, we end up with this prototypical gene structure. So this is what genes would look like in prokaryotes and eukaryotes. So here we have prokaryotes. They have a promoter, they have a terminator, and they have the gene in between. Very simple arrangement. And then in ourselves, eukaryotes, we have promoters, we have terminators, and then we have the gene in between. But our genes are interrupted by introns, which are these beige areas, which have to be then removed in the RNA before the purple areas are concatenated back together. So a more sophisticated hierarchy. Translation itself is a very complicated creature, and it involves many moving parts. But the important thing to understand is that the ultimate goal is to put together the building blocks of proteins in the right order so that the protein has the right function. So amino acids are the building blocks of proteins, and the amino acids are joined together on the surface of a ribosome, but based on the instructions received from the messenger RNA. So the messenger RNA brings in the information, and the ribosome simply obeys the instructions, working alongside tRNA to form the polypeptides. Your understanding of translation can be elicited by asking you questions on the genetic code. And the genetic code is, how does the ribosome know which amino acid to add next to a growing polypeptide chain? So we call those regions on the nucleotides, the RNA, codons. And the codons are then read by a translation machine, which is quite complex, and we'll learn that in future, that brings in the appropriate amino acid. Unfortunately, the messenger RNA doesn't always begin at the right same place every time, but it does begin further along at a position called a start codon. So start codon is nested inside the messenger RNA, but it is a special place where the ribosome then knows what to do, and it starts looking for the first AUG sequence along the messenger RNA, and when it finds that, then it puts in the first amino acid. And the first amino acid is always going to be methionine, because AUG stands for methionine. So here we have a slide that gives us an overview of transcription here between DNA and messenger RNA, and translation, a step that takes messenger RNA and then converts that to a polypeptide. You can see methionine is the very first amino acid to go in. So one of the things you'll learn is the details behind this step-by-step manufacturing process. And in another couple of chapters, chapters 8 and 9 I believe, we're going to have to learn this in very, very fine detail. Just like the front end of the messenger RNA has a very special place where the first amino acid is put in by the start codon, along the length of the messenger RNA there's another sequence called the stop codon. And the stop codon tells the ribosome when to stop manufacturing further polypeptide. So we're going to have to learn that too. We spoke about the tRNA as being a very important component of the manufacturing process for polypeptides. Well, the tRNA is made of RNA, And a certain region of that tRNA contains an anticodon sequence of nucleotides. And those anticodons are then matched up physically with the codons on the messenger RNA on the surface of the ribosome. Again, sounds sophisticated, but in reality it's a very simple process. So when we finish, we'll understand that the genetic code carries specific instructions for which polypeptide to make based on this table here. So this is a deciphering of the biochemical mechanisms that most living things, 99.99%, use for making polypeptides. You don't have to learn this table, but you do have to learn certain features about this table in the future when we get there. So you can see here, these are the 20 amino acids. We're going to have to learn their 3-letter abbreviation. We're going to learn their 1-letter abbreviation by the end of the course. But we don't need to know every codon that specifies that amino acid. But we do need to know that some use 6 different codons to make the same amino acid come into the ribosome. But others, they use 1. So that pattern has to be learned. Next, we're going to look at some commonly used techniques that have helped us in the past and currently in deciphering genetic information. Since the DNA molecule is the same amongst all living things, the only difference is the information that it carries in neighboring nucleotides. So I may have an AAG, you may have an AAC at the same location. So how can we determine what I have versus you? Well, the mechanism of doing that has an origin. And the first place to start is to be able to separate DNA molecules based on size. So gel electrophoresis is an experimental technique that allows us to do exactly that. The same technique can be applied to separating proteins, but that's a bit more complicated. So separation based on size is the major hallmark of gel electrophoresis. But what you may not know is that you can also separate DNA based on shape other than just size. And also you can separate molecules like proteins based on their charge. So it's a pretty universal technique that we need to understand. The technique begins with somebody making a gel. So agarose or polyacrylamide is heated in the microwave till it melts into water, and then that solution is poured into a tray, and the tray has a comb on one end which will form the wells into which the specimens are dropped. The gel is allowed to solidify, the comb is removed, and hopefully the wells stay intact, and then the gel is taken and placed in a tray, and the tray contains an electrolyte, a solution that can conduct electricity, normally sodium chloride or something like that. The electrodes are then charged with a power supply, and the power supply places an electric current across the gel once the DNA has been loaded or the protein has been loaded. Once the electric current is applied, because DNA is a negatively charged acid molecule, it will be pulled towards the positive end of the tray using attraction. And the speed at which the molecules move in a very simple experiment could be based on size. So here, the smaller size molecules find it easier to get through the gel than the larger size molecules, and therefore we can separate DNA in each one of these lanes according to size. Once the DNA has been run on the gel, it cannot be seen with the human eye. So what we tend to do is to place other chemicals either into the gel, or we soak the gel in that chemical afterwards, and that chemical then binds to the DNA, and under appropriate lighting conditions, the size of the bands can be visualized, can be seen. And from that we can then identify what band is large and what band is small. Most of the time we also include fragments of known size in one of the lanes so that we can have a marker. So maybe the first lane would contain multiple size fragments of different sizes that are given to us by a company. So the fragment down here may be running at 200 base pairs, the fragment here will be running at 500, and maybe the one up here will be at 1,000. Then we know that our fragments are just about 1,000 base pairs in length compared to the smaller ones. So that's how a size marker works. Once the gel has been run and visualized, it's not possible to keep these gels long term because the DNA tends to drift based on physical diffusion all over the place. So one way to securing a copy of that DNA is to move it from the gel onto a piece of synthetic membrane. And that process of transferring the DNA out of the gel onto the membrane was invented by a gentleman with the name of Southern, so he gets credit for that. So that process, if it involves DNA, is called Southern blotting. If it involves RNA, the scientists simply thought of the opposite and called that Northern blotting. And more recently, you can do the same technique with proteins, and therefore, to complete the compass, somebody came up with the clever idea of calling it Western blotting. There is no such thing as Eastern blotting at this time. Figure 114 shows us two gels. This gel is a DNA gel. There's your marker, and these are your samples that were run by the scientist, and here is another marker. So you can see, if you know the size of these bands, you can then estimate the size of the bands in lanes 2 to 7. This is a gel of protein, and you can see here, this is a marker. The size of these protein bands is known, and then this is your unknown samples being run. The thickness of the bands corresponds to the amount of protein. So this band contains much more protein than this band here. So by looking at this, you can tell the relative abundance of different proteins in this sample. Gel electrophoresis has now evolved into a very, very sophisticated technique that involves very little gel and is performed by robots and computers. So because of that advance in technology, we now have the ability to quickly and cheaply sequence DNA from different organisms and species. We can not only sequence small segments of DNA, we now have the capacity to sequence the entire genome of that organism very quickly and cheaply. Because of that, the field of genomics has become enormous. Genomics provides a more complete picture of what's happening at the biochemical and cellular level inside an organism or inside a cell, such as a cancer cell. So by sequencing, interpreting what we have sequenced, and by comparing those sequences with different genomes of different organisms, we can learn the functions of that region of the DNA. The success of genomics has led to the mushrooming of other sectors of the biochemical analysis of molecules of life. So proteomics is the study of the complete sets of proteins in a cell or an organism. The transcriptomes are the complete set of messenger RNA available in that cell. Like we said to you earlier, not all genes are active, and different genes may be active at different rates. So this gives us a relative abundance of some of those messenger RNA molecules. So they may be different in a cancer cell compared to a normal cell, and by comparing the two, we get insights into what's happening inside that cancer cell. Metabolomics allows us to study just the chemical processes involving that particular cell, tissue, or organ, and even an entire organism, so we can get a better understanding of the biochemistry. By studying the different omics, we're getting a fuller picture of how life processes work, rather than in isolation as was done in the past. The final part of the chapter goes and talks about evolution and how evolution has a genetic basis. What we would like you to gain from this is the full circle of knowledge about how evolution and genetics are intimately tied together. Evolution may seem to you like a very complicated process with many parts, but at the top of the triangle, the pinnacle, is one single statement. If the DNA of an organism changes in its population, then there is evolution. That's it. Because DNA is subject to mutation, change, chemical change, that can lead to new combinations of alleles and new functions of existing or new DNA. And all that leads to changes in the organisms that acquire those mutations and therefore differences between individuals of that species. And this applies to humans also. The impression that we've been left with by Chapter 1 is that the biochemical processes that replicate DNA are pretty ancient, and the central dogma that allows that DNA to be expressed is also universal amongst all living things. So if DNA acquires mutations, then it will acquire mutations in all living things, and that can lead to changes between individuals. One thing that we'd like you to not forget is that life on Earth is pretty old, and we believe, without any exact information, that sometime between 4 billion years ago and 2.5 billion years ago, life began on this planet. We do have fossils that go back sometime to 2.5 billion years ago of single-celled creatures that look like those that exist today, such as these, which are thought to be the fossilized remains of 3.5 billion year old Archaea from Shark Bay in Western Australia. The theory of evolution, as originally defined by Charles Darwin back in the 1850s, and also credited these days to similar work done at the same time by Alfred Wallace in Indonesia, suggests that living things, DNA, and evolution all go hand in hand. If you understand evolution, you will automatically understand every aspect of biology, including genetics. The definition of evolution, given in Chapter 1, says it's the scientific theory that all organisms are related by a common ancestry and have diversified over time. So let's put this in context. We learned earlier that life started with some common ancestors, and they devised mechanisms of copying DNA and then translating that DNA into protein through transcription. So if that's what happened, then we know the basis of this statement here by common ancestry. So those biochemical processes may not have changed much. So that gives credence to the age of living things. But the diversified part tells us that these mutations have increased in different branches of the tree of life, therefore giving rise to different forms and different functions, therefore generating new species and new sets of organisms. Charles Darwin looked at evolution from the natural selection perspective. That is, that organisms compete for resources, and those resources favor one form over the other. But looking at what causes those different forms, the long neck versus the short neck of a giraffe, we now know that that phenotypic difference in giraffe neck is all due to genetic differences that the giraffes inherited from their parents, as well as some environmental impact. Therefore, we can now tie together the genetic component, the DNA component, the phenotypic component, and the definition of evolution. Thus, Darwin's statement, survival of the fittest, means that those creatures that have the appropriate alleles will have a greater chance of surviving to reproductive success, and therefore passing on those alleles to future generations. Evolution itself has 4 subdivisions, 4 mechanisms that can bring about changes in the frequency of alleles and genes in populations. Natural selection we just spoke about, that's the success of leaving behind babies, called differential reproductive success. Migration is a force of evolution, because individuals from other populations carrying different alleles may come into your population, therefore changing the frequency of alleles. Number 3 is mutation. Mutation is generally a slow process that introduces new alleles into the population that can be tested and challenged. And then lastly, the one that students generally tend not to remember is we have random genetic drift, and this occurs when the population size drops to low numbers, or when mating behavior in individuals of that sexually reproducing population is no longer random. Once we start looking at information beyond a single individual or a single cell, we enter the realm of population genetics. So population genetics looks at relationships among organisms. And those relationships can then be used to generate trees, relationship trees based on RNA, DNA, or protein differences. And that can result in diagrams such as this, which show you the computer's mathematical relationship generating function, where it puts together closely related organisms and separates more distantly related organisms. Not only that, if you have the right information, you can also generate the length of these arms proportional to time. So here will be a time factor. So this will be a very ancient common ancestor, and then this will become more recent, and this will be the present day. So you can see that the medium size and the small ground finches are more closely related to each other than they are to the large ground finch. And that's the way to read these diagrams. If the computer can be supplied with information from a species that doesn't have the feature that you're interested in, then all the better. So that would be called an outgroup. So let's go here and see if we can find an outgroup. There is no outgroup, right? So in this case, we don't have an outgroup. All these are related to each other, and they are discussing the feature or comparing the feature that we're interested in. An outgroup in this case could be another species of finch that has nothing to do with these. Just an added in-depth feature that you must understand is this word here called homoplasy. Homoplasy. Homoplasy simply means that two branches of the tree of life that are not related directly to each other but have something in between, the two branches may develop a common feature, characteristic like wings, independently of each other because both branches need access to flying. So those we call homoplasy. Clades are simply groups of organisms that share certain features in common compared to their more distant neighbors. So here we have humans and primates, gorilla example, and one thing they share that the wolf doesn't have would be opposable thumbs. If you look at the foot of a wolf, its thumb cannot be used to touch its fingers, whereas humans and gorillas can do that. And nowhere else down this list is that feature available because it only evolved along this branch of the tree of life. So these two would form a group called a clade. Now if you go back further and then you come to this point, anything prior to this doesn't have a placenta. So the baby doesn't form inside a placenta. But what these three do share, and wolves share with the primates, is to have babies that are live born, and those babies are born through a placental interaction. So that will form another clade, but then we have to define that as the placental clade. And going back further and further, you can see different features evolved at different points in the history of life. And the further back you go to the left, the more ancient those particular relationships become. Why do we have to learn about phylogenetic trees? The answer is simple. It appears that they are the most accurate way of deciphering the past. They're more accurate than looking at fossils. They're more accurate than looking at developmental features or morphology. In fact, they are the most accurate tool that we have. And the premise is very simple. As I said earlier, those species that are more closely related will have the smallest number of differences in their DNA, RNA, or protein. In fact, once you understand how fast a particular region of DNA mutates and changes, you can then use that to generate a timeline of how much in the past and the speed at which these genes have changed between two species. The last two slides in this chapter simply give us an example of how to understand these phylogenetic trees. So in the example given here, we are going to look at 7 different species and a small region of a gene responsible for making hemoglobin. And we're not going to look at the entire gene, we're just going to look at 15 nucleotides. So here are the 15 nucleotides. We let the computer software rearrange all the information, and it generates this order for the 7 sequences. So sequence A, from whichever species that came from, and species B, they don't differ at all between these 15 spots for the beta globin gene. So the computer groups them together near the top. Next, out of the remaining 6 sequences, the computer looks for the sequence more closely related to these. And it finds that C is more closely related to A and B. So it puts sequence C next. And then it does the same rearrangements with D, E, F, and G. And as you can see, G, according to the computer software, is the most distantly related compared to A or B. So this rearrangement is based on a tendency to be more and more different as you go down. The remaining panels of this figure simply break up the information into chunks. So what the authors are saying is that sequence A, B, and C are more closely related to each other. Therefore, they may have something going on just in their common ancestor that led to these 3 creatures having the information that they carry. So that would be a clade. So if you're not sure what a clade is, go back to this figure, and you can see in this case these 3 will form a clade, or these 4 will form a clade. So likewise, rather than looking at morphology, we're now looking at sequence, and these 3 would form a clade. In fact, A and B would form another clade, which is a subclade of clade A, B, and C. Regardless. So what does that mean? Well, that means that the computer is going to tell us the probability of mutations taking place. So the more ancestral the sequence, the more similarity or difference it would have with our top rated. So here the difference between D and the other 3 above is quite large. So D can be considered an outgroup, an anchor, so that we can get the relative positions of A, B, and C to each other, and the time that it took for A, B, and C to become different from D is also suggested by the software. So a long branch here led to the evolution of these 3, and that mutation there allowed C to break away from A and B. So that's one way to interpret it. As the computer expands the study to include more and more sequences, then the branches are added in according to how much time it thinks it would have taken for these differences to materialize if the rate of mutation was steady. And that's the key. The rate of mutation is assumed to be steady between the different sequences. That may not be the case in reality, but so far it works for us. And finally, once you take this tree and then you compare it to known morphological or biochemical previous understanding, you can see that the 2 are more or less in agreement. Students need to be competent in understanding the underlying basis of phylogenetic trees, as well as working through some examples of its application. Thank you so much.